Welcome to the channel. In today's video, we'll explore one of the most fascinating tools in modern brain imaging, Diffusion MRI, and learn how it helps scientists visualize the brain's white matter in 3D. Our brain is made up of two main types of tissue: gray matter, which contains neuron cell bodies, and white matter, which contains long, myelinated nerve fibers that connect different brain regions. About 45% of the brain is white matter, and these fibers act like the information highways of the brain, constantly sending signals between different areas. Diffusion MRI is a special type of MRI that allows us to see how water molecules move inside the brain. By studying that motion, we can indirectly map the structure of these white matter pathways. Inside our brain, water molecules are always moving randomly. This is called Brownian motion. But in white matter, this movement isn't completely random. It's restricted by the structure of the nerve fibers. Imagine water flowing freely in a glass of water. That's isotropic diffusion. Now imagine water moving through a bundle of straws. It can move easily along the length of the straws, but not across them. That's anisotropic diffusion. From DWI to DTI, the process begins with a special MRI scan called diffusion-weighted imaging, or DWI. Multiple DWI images are captured, each with a different direction of diffusion sensitivity. Sometimes 12, 41, or even more directions. Each of these images gives us information about how water moves along that specific direction. By combining all of them mathematically, we can estimate something called the diffusion tensor. That's why it's called diffusion tensor imaging, or DTI, understanding the diffusion tensor. A tensor is just a fancy mathematical word for a 3D object that describes direction and magnitude. In DTI, each tiny voxel of the brain, think of it like a 3D pixel, contains a tensor that represents how water moves inside that area. If the tensor looks like a perfect sphere, diffusion is equal in all directions. That's isotropic, like in gray matter or cerebrospinal fluid. But if it's stretched into an ellipsoid, it means water prefers to move in one direction. That's anisotropic, typical of white matter fibers. So, scalar maps. From these tensors, we can calculate useful measurements called scalar maps. These are single-value summaries that tell us more about the tissue. The most common ones are trace, or mean diffusivity. Measures the overall amount of diffusion, regardless of direction, useful for studying tissue damage or stroke. Fractional anisotropy, IFA. Measures how directional the diffusion is. High FA means very directional, typical in white matter tracts. Low FA means random diffusion, typical in gray matter or damaged tissue. These maps help doctors and researchers study how brain connectivity changes in disease, development, or injury. Tractography. Mapping the brain's highways. Once we know the main diffusion direction in each voxel, we can start connecting them, like tracing roads on a map. This process is called tractography. Tractography algorithms follow these diffusion directions step by step, reconstructing long 3D curves that represent the brain's white matter pathways. These colorful 3D models show major structures like the corpus callosum, which connects the left and right hemispheres of the brain, or the cingulum bundle, which links parts of the limbic system. This gives neuroscientists and doctors a powerful visualization of how different brain regions are connected. Tools for diffusion MRI analysis. To perform diffusion MRI analysis, researchers often use 3D Slicer, an open-source medical imaging software. With 3D Slicer and its extension called Slicer dMRI, you can load diffusion data, calculate tensors, create FA and trace maps, and perform 3D tractography, all in an interactive environment. So, to sum it up, diffusion MTeam R.4 gives us a window into the brain's wiring system, allowing us to map the hidden highways of information flow. It's a powerful combination of physics, mathematics, and neuroscience, and it's helping researchers better understand brain development, injury, and disease. In the next video, we'll move from theory to practice and actually build diffusion tensor maps and tractography in 3D Slicer. So stay tuned and don't forget to like, subscribe, and hit the bell icon if you'd like to see how this works hands-on. Thanks for watching, and see you in part 2.